shared, and gosh, I need to share my screen, don't I? Hold on, let me share my screen. All right. Okay, so to continue where we left off, we were talking about structure, protein structure. We talked about amino acids already, and then we started talking about the formation of secondary and tertiary structure and some characteristics of that. So let's recap together. You can unmute yourself. Preferences better than typing in, otherwise you can type as well. What is the role of hydrophobic residues in the folding of a protein? Who remembers or who can summarize that?
They usually want to be away from the aqueous solution that the protein is in, and so will be in the middle of the fold, so of the tertiary structure.
Okay, so the hydrophobic residues drive folding because proteins usually in cells exist in aqueous environment, and those hydrophobic residues don't want to interact that much with water. So they start to pull towards each other, then the protein starts to fold. But what are the main forces that stabilizes the secondary structure? So we go from primary, and we start getting some hydrophobic interactions that pulls the residues together into a folded state. However, when the secondary structure is forming, it gets stabilized by what force?
Hydrogen bond?
Yes. So hydrogen bonding is then stabilizing the secondary structure. So hydrophobic residues drive the folding. However, when the protein starts to form secondary structure, the water is kicked out of the interior mostly. And instead of having hydrogen bonding with the water, the internal residues start having hydrogen bonding with each other and start to stabilize the secondary structure. But we still have a lot of free energy, and we need to contain that free energy. So the protein starts to also form a tertiary structure. So other than secondary, it starts to contain more of the energy by certain forces. What other forces take place in stabilizing a protein's tertiary structure?
Disulfide linkages?
Yes, it is the strongest bond. So if we have sulfide groups, if we have cysteine residues, yes, we form disulfide linkages. Not all proteins contain an abundance of cysteine, so not all proteins form disulfide linkages. Okay, so we know that we have hydrophobic interactions. We talked about that. We know that there's hydrogen bonding, potentially disulfide. What other bonds contribute to the stability of the tertiary structure?
Dipole interactions, they're weak, but they're additive.
Yes, absolutely. Dipole, or we call them also van der Waals interactions. So between neutral atoms and neutral molecules. So you get that interaction that is additive. It's weak, but additive. There's one more.
Ionic.
Yes, of course. And ionic interactions can be attractive or can be repulsive. So sometimes they aid in the stability, but through attractive forces. But depending on the pH, if the pH causes more of the same charge to happen, that can cause protein unfolding by breaking down ionic interaction, causing more of a repulsion. So when we talk about stabilization of a tertiary structure with ionic interaction, we're talking about attractive ionic interactions. Okay, so we introduced enthalpy and entropy, and I know it's not a really a favorite topic, but if we want to simplify it, what's enthalpy?
Heat flow.
Heat flow, it's actually a heat contained. So the energy contained within a molecule, within a protein molecule that is stable. So whenever bonds are formed, we have contained the energy. And in order to denature the protein, so when we run DSC, for example, some of you are familiar with that. We're going to introduce it later on. When we run differential scanning calorimetry, we introduce a heat flow and the protein denatures. So the heat that we introduce should equate the contained energy that stabilized that protein. So it's the contained energy. Now the entropy, on the other hand, is what?
Disorder in the system. The
Disorder. Yes, it's the chaos. So it is the opposite of potentially enthalpy. So basically when you have bonds formed, you have contained energy. And when you start breaking these bonds, you start causing free energy to increase and chaos. So a couple of hydrogen bonds, for example, can cause a great entropy, because when they break, and then there will be a distance between the molecules, other bonds are going to break, and it becomes kind of additive process and leads to spontaneous denaturation. So you just want to remember that delta G of denaturation equals delta H, which is the change in enthalpy, minus temperature, and delta S, which is the entropy or the chaos. So when you break few bonds, you have a great change in entropy. So the delta S becomes large, then delta G of denaturation becomes negative. And when it's negative, that is a spontaneous denaturation. Okay. Things that you also want to remember are the solvents that disrupt these interactions. So if you want to disrupt hydrogen bonding, what could you do to disrupt hydrogen bonding within a protein?
Could add detergents.
Detergents, yes, such as urea, SDS, sodium diacetyl sulfide, and these will break hydrogen bonding, and they also break hydrophobic interactions. Hydrogen bonding is also weakened by heat, whereas hydrophobic interactions are strengthened by heat. Okay, what breaks disulfide linkages?
Mercaptoethanol or reducing agent.
Yes, which is a reducing agent. So you can add beta-mercaptoethanol, DTT, sulfides, sometimes even cysteine, just the amino acid cysteine, can act as a reducing agent. What about ionic interactions? How would you?
Acid or bases?
Say that again, Nigel. What did you say, Nigel?
I think he's muted.
Can you hear me? Nigel, what did you say?
Oh, sorry. I think there was something wrong with my mic. I said acids or bases, depending.
Yes. So pH, changes in pH. So when you change the pH, you can disrupt ionic interactions. Also presence of salt. Depending on the type of salt and the concentration of salt, you can either enhance ionic interactions or you can disrupt ionic interactions. Okay, great. This is all good information to keep in mind as you study, because then it, as we talk about structure later and function, we're going to all come back to the, what is the secondary structure of the protein, what's the tertiary structure, what the bonding means. So it's all good information. We're building a puzzle as we go. Okay, so how does the free energy concept explain the folding? I kind of already talked about it a little bit, because when you form a peptide, a protein primary structure, which made up of the amino acid sequence, there's a lot of energy. So the protein starts to fold on itself to contain the energy by forming interactions to reduce the free energy to a very stable form. And sometimes it has to be a quaternary structure to maintain stability. So it's not always just tertiary structure. We'll talk about that later on today. So again, I'll keep an eye on the time so that I can give you a break before it's just too much information to process. Also, if you want me to repeat any concept and if I'm going too fast for you, please just, feel free to interrupt and ask. Do you have any questions from previous lectures? I know Paul asked a question, which is good. Any other questions?
I have a question.
Oh,
perfect.
Okay,
We have Nigel and Chrissy. Go ahead, Nigel.
Okay, so I just have a question about the interactions within a peptide chain.
Yeah.
I was wondering, between two amino acids, is it possible to have more than one interaction, like let's say hydrogen bonding and dipole interactions occurring at the same time?
Yeah, it's possible. Definitely, if there is if there are the right atoms and the right charge, and also the right distance between these two amino acids, and it also depends on their R group, because R groups can cause steric hindrance, which might impact how the molecules and the bonds can form and how the folding can occur. So it all depends, but yes, more than one interaction between two amino acids can occur. Yep. Okay, good question, Chrissy.
Yeah, so regarding transglutaminase, I just kind of realized that your example was for bread made with rice flour.
Yeah.
So I don't really know that much about rice, but is there enough protein in it for that to make a significant difference, or would adding other proteins like protein powder or something help?
Yeah, definitely adding other protein help, but if you just want to make a gluten-free bread, for example, so you would be limited to what types of grains you can add and flour you can add, and not. It's kind of like this unique viscoelastic property is unique to gluten, and we haven't found a protein that has similar characteristics to form that loaf. So you really need a nudge and help, and the polymerization through transglutaminase can help that way.
Thanks.
Okay, any other questions or comments? Okay, good. So we'll just move on then. So we talked about the primary structure and the potential folding. I want to dive into a little bit the secondary structure. So our different secondary structures that can form. You have alpha helix, beta sheet, beta turn, random coil. So all of these are under the category of secondary structure. So alpha helix is just as the name indicate, you form a helix. And you need a specific, for the most part, theoretically, you need a specific sequence of amino acids. This sequence here, P and N, stands for polar, nonpolar. So a sequence of polar, nonpolar, then two polar, then two nonpolar, followed by the same sequence, again, polar, nonpolar, two polars. This is kind of the general sequence that would potentially lead to the formation of an alpha helix. There are certain number of atoms in one loop. So in formation of this one loop here, there are 13 atoms. So this is a loop. This is a loop. So the formation of one loop, you have 3.6 amino acid residues and about 13 atoms within these 3.6 amino acid residue. And this is an average. It's not like every alpha helix is exactly that way, but this is theoretically the constitution. So, and this here is the axis of the alpha helix, and you have formation of hydrogen bonds between the different residues and functional groups within the amino acids. The hydrogen bond usually are parallel to the axis of the alpha helix. So you have hydrogen bonding occurring in parallel to that axis, basically. And in most cases, your hydrophobic groups, it could be just the R group of the amino acid itself, would be targeted towards the interior of that alpha helix. One thing I want you to remember is the presence of proline. So the presence of proline is disrupts the secondary structure. That means disrupt the formation of an ordered structure. So if you disrupt alpha helix formation, disrupt beta sheet formation. So instead of forming a structured form of a secondary structure, you have something we call random coil. So the presence of proline in the sequence limit the flexibility of the folding and the formation of an orderly structure due to this bond, which is phi here. So the bond between the supposedly free amine is no longer free because it is your R group here is linked to it. So then you don't have that flexibility anymore in turning, orienting, and formation of certain interactions to form an alpha helix or a beta sheet. So I point it out because later when I talk about caseins, for example, I tell you, well, proline is about 17% of the composition of casein. So for casein, you see very little amount of alpha helix and beta sheet and a lot of random structure. And it does have, because of that, an open structure rather than a globular structure as well. So this is important later on when we talk about functionality of the caseins. So beta sheets, so it's basically sheets as you can see here in the structure. And within one molecule, one protein, strands within the one protein can form in a parallel formation. So this strand and this strand, for example, they just come from one string. So if you have, say, a string, you can form it, you can form parallel sections within that string. So when that is formed, then you have hydrogen bonding perpendicular to the axis. So you have hydrogen bonding forming that way. That if you can see here, there's this red structure indicating hydrogen bonding. So they are perpendicular to the axis of the beta sheet formation. Usually, the sequence is alternating polar, nonpolar. So when you have a sequence within the protein that has this alternation, then it would most likely form beta sheet. Segments within the proteins, here specifically rich in valine and isoleucine, have a tendency to form beta sheet. Okay, it's not something that you really have to remember, the valine and isoleucine, but it's just FYI. But I would like you to know that this is the theoretical sequence, and the hydrogen bond is perpendicular to the axis of the sheet. Beta turn is another formation, and it's just a turn as it is described here. And there's your hydrogen bonding here that stabilizes this beta turn, involving about 4 residues. And these are some of the amino acids residues that may be involved in such a configuration. And sometimes when upon processing, heat processing, or like in dough formation, for example, some of the beta sheets reverse and form beta turn. So sometimes when you process the protein and you're looking at the secondary structure changes, we see an increase in random coil sometimes a conversion of alpha helix to beta sheet, and sometimes you see conversion of beta sheet to beta turn. Now, why is all of this important? Because it's impacting the structure. And later on, when I talk about function, I'll link I'll link some of these structures to the function. Okay, so one thing, for example, here that relates things together is denaturation temperature. So you see that some proteins have lower denaturation temperature than other proteins. If we want to look at the makeup, we see this is mostly alpha helix secondary structure into a globular shape. And this is why protein beta lactoglobulin, sorry, you have a lot of beta sheets and one alpha helix and some random coils and turns. You have some turns here and here. Okay, so this denatures at higher temperature. So the secondary structure also imparts certain stability, and the beta sheet is more stable to heat than alpha helix. So it resists denaturation more than alpha helix. So oftentimes, let's say when we do FTIR analysis to look at secondary structure, we can look at alpha helix to beta sheet ratio, and it will tell us if the protein have seen some denaturation, because the alpha helix to beta sheet ratio changes upon processing. Some of the alpha helix structure convert, and you end up having beta sheet. But it not in all cases. It also depends on the sequence of the amino acid. Sometimes the alpha helix converts to random coil, not necessarily to beta sheet upon heat. Okay, so again, moving from secondary structure, we still have free energy, so the protein will form its tertiary structure. And more bonding occurs other than hydrogen bond. We have hydrophobic interactions, we have disulfide linkages, we have the ionic, and we also have the neutral, which is the van der Waals. Once all of these interactions occur, all of the free energy gets contained, then you get a stable tertiary structure. So what impacts the tertiary structure? So when we say configuration of tertiary structure is a function of what? What impacts whether it's going to be a globular or it's going to be open structure or it's potentially going to form a quaternary structure for stability? What do you think is the main driving force?
Like the environment?
Environment is important,
pH, yeah.
But before the environment, if we want to talk about something intrinsic, not extrinsic.
The side chains?
Yes, so amino acid composition and sequence, not just the composition. Composition definitely impacts how many hydrophobic groups we have, how many ionizable groups and all of that, but where are they in the sequence? That would promote certain interactions. So configuration is a function of amino acid composition and sequence. Okay. So tertiary structure is supposed to be very stable, but the composition and sequence would either result in an open structure like the beta casein, for example. Remember that has high percentage of prolines, so it has a secondary structure breaker in it. So you don't have that much flexibility. 17% of the composition is proline. Then you have this naturally open structure because of limitation and steric hindrance and inability to form a more compact structure. Whereas beta-lactoglobulin and other proteins we'll talk about later in the semester have this globular structure, have really ordered secondary structure. So you rarely, you don't have that many random coil. You do have the alpha helix and you have a lot of beta sheets, and you have flexibility to form a globular protein in a way where you hide most of your hydrophobic residues in the interior. So beta-lactoglobulin is a unique protein, has ordered secondary structure. Most of its hydrophobic groups are on the interior and makes it a very, very soluble protein. because it has very low surface hydrophobicity compared, for example, to beta-casein, where it has an open structure and a lot of the amino acids are forced, the hydrophobic, to be exposed to the environment. So the protein structure doesn't end with secondary structure, a tertiary structure. Some of the proteins are stabilized in a quaternary structure. The most classic example is your casein micelle. So if you look at the casein micelle, this is a kind of a model. It's later on when we talk about dairy proteins, I'll show you the newer models of the casein micelle. But here is just to visualize that the casein micelle is made up of multiple proteins. It's made up of 4 different proteins, alpha S, alpha-1, S2, and beta casein, and kappa casein. So they interact via these blue dots here are calcium phosphates. So they interact together via calcium phosphate bridges. So you have these electrostatic type of interaction or ionic interactions. And also they have hydrophobic interactions in the interior that keeps them together. And then on the exterior, you have submicelles that have a lot of kappa casein. So the high percentage of the kappa casein stabilizes the micelle because it has this glycomacropeptide in it that is glycosylated and gives a negative charge. So you have a really stable micelle with kappa casein mostly on the outside, and the other hydrophobic caseins are interacting inside, and also you have the calcium phosphate bridges. So this is a very classic quaternary structure that stabilizes the casein micelle within milk, and they don't sediment unless you get hydrolysis and overstorage. You do get that with enzymes, endogenous enzymes, and bacterial growth, microbial growth. Again, we'll talk a lot about dairy proteins later. Okay, this is a quaternary structure of a globular plant protein. This is specifically illustration of a globular protein in soy that associate as a trimer. So each of these colors represent a singular protein, and they associate with each other via hydrophobic interactions. So because the individual proteins have high surface hydrophobicity on the surface, they have high percentage of hydrophobic amino acids. So they want to hide from water, so they associate with each other and form trimers and stay stable that way. Okay, so different quaternary structure have different association, and the bonds that take place either hydrogen bonding is involved, ionic bonding, hydrophobic, and sometimes you also have disulfide bonds that stabilize a quaternary structure, and we'll learn more about that when we talk about legumes, soy proteins, and other legumes. And these interactions are obviously impacted by pH and the salt content or the ionic strength. So in certain situations, these quaternary structures are destabilized based on the extrinsic factors, pH and salt. We'll give plenty of examples based on those different types of proteins later, but just keep in mind that the extrinsic factor impact the stability of these quaternary structures. Okay. So even the quaternary structure tend to either be globular or fibrous. So if you look at the collagen, for example, it forms fibril type or structure. And if you look here, it's the hemoglobin, it forms a globular quaternary structure. So this structure that I showed you here, that's a globular quaternary structure, okay? So different proteins, when they form quaternary structure, they're either a globular structure or fibrous. Those that form fibrous structures are high in percentage of amino acid. polar amino acids. So then you have high percentages of interaction with water. So they're actually, this fibril shape is basically twisted polypeptide chain that have linear aggregation of small globular proteins. And then they form this fibril type of structure due to the presence of high polar amino acids. They form as structural proteins. And because of the high polarity, they stabilize mostly by hydrogen bonding, and they do have cysteine in them, so they get stabilized by disulfide linkages. So the globular type, which we will be talking a lot about, we have a lot of different types of proteins. Plant proteins are mostly globular. The functional components of the plant proteins are globular. So we'll talk a lot about them later. And they just form a quaternary structure in a globular shape that, like I just showed you in the former slide. Enzymes are mostly globular as well. So we'll have examples of globular protein structures when we start talking about the different proteins from different sources.
I have a question.
Yes?
Is it I don't know if it's a fair assumption to say, but you're talking about how the plant proteins are pretty much globular, and then for the most part, would animal muscle proteins be more the fibrous? Is it pretty easy to make that distinction between animal versus plant proteins as to what their quaternary structure is going to be?
Yeah, so animals, specifically meat, yes. But when we talk about animal-based, like dairy proteins, you don't have that fibrous structure. So whey protein is globular, but it doesn't form a quaternary structure unless the pH is around its isoelectric form point. So it kind of ate whey protein or beta-lactoglobin associate together because of the pH change. But no, we cannot just say all animal-based are fibrous. It's really the meat proteins mostly are fibrous.
Yeah, but animal, the animal meat ones could we say that all the animal meat proteins are fibrous? Would that be right?
Yeah,
okay.
Yes. You form quaternary structure that is fibrous.
Okay.
Yeah. So you want to think about the muscle. All the muscle that comes from the meat, you have that fibrous structure. And that's the challenge when they want to make meat alternatives from plants. How can we make these proteins aggregate in a way to form fibers? So that's something we talk about later on, but yeah.
Thanks.
Good. What else? Anything else? All right. So last bit here, or before the last bit about the proteins structure formation, you have homoproteins. That means you have proteins that are only made up of amino acids. You don't have any other type of molecules associated with the protein. But there's sometimes you have proteins that have other groups like phosphates in them, which is becomes a phosphoprotein, such as alpha and beta caseins in milk. You have glycoproteins, such as kappa casein or visilin and conglycinin, for example, in legume proteins. Some of them are glycosylated, so then we call them glycoproteins. So the casein micelle is stabilized because of the presence of phosphoproteins and glycoproteins. So the phosphoproteins cause these bridges, and the glycoproteins causes the stabilization of the micelle on the exterior. We have lipoproteins in eggs. We'll talk about them when we talk about egg proteins and their role in emulsification. So now the last bit about the protein structure is the net chart, which is really important and it impacts a lot of functionality. So, every amino acid can be either charged or positively charged or negatively charged, depending on the pH. But when you form the polypeptide, then you have different amino acids, different charges, then some charges cancel each other and some charges don't, so you end up with a net charge. So the net charge is basically the charge of the protein after cancellation of the pluses and minuses. And that depends on the amino acid composition, how much basic amino acids you have versus acidic amino acids, and also depends on the pH. So you can actually change the net charge of the protein based on the environment. So some proteins have an isoelectric point around neutral. Some proteins, or most of the proteins we work with later on, you'll see have an isoelectric point in the acidic range of 3.5 to 5.5 range. So what that means, the pH at the isoelectric point, meaning that at that particular pH, all charges cancel each other. So you don't have a net positive or a net negative. If you change the pH above the isoelectric point, you start to add negative charge. Why? Because then above the isoelectric point, you start losing the protons, because now you have more basic environment. Then protons from the different amino acid will be dissociated. Then you end up having more net negative charge. Whereas on the other hand, if you put the protein in an acidic environment, so you have a lot of protons in the environment, then the protein starts picking up protons, and then it becomes net positive charge. So then you have more interaction with water and less protein-protein interaction. So this means your protein, when you have a net charge, whether a net positive or a net negative charge, then you have better solubility. Salt also impact the charge in a way that we'll talk about salting in and salting out later, but sometimes a little bit of salt can interact with the surface of the protein and add kind of like a charge load. Then you have more water interaction. However, different types of salts have different impacts, and different concentrations of salt have also different impact. At higher concentration, then the salt interacts strongly with the protein and compete for water interaction. Then you end up having salt-water interaction and less protein-water interaction, because you are also shielding charges. With salts interacting on the surface of the protein, then you shield the charges, and then you no longer have water interaction. Instead, the proteins start interacting with each other via hydrophobic interactions and ionic interactions. So the environment affects your net charge and interactions with water, whether it's pH or salt. Let's think about this for a minute, because this will remind you that you want to know which amino acids are charged and what charge they carry. And it reminds you that the composition of the protein impacts the net charge at a particular pH. So let's think about this question in red. Any results? So at around neutral, what do you think the charge of lysine is? Is lysine a basic or acidic amino acid?
Basic.
Basic. So at around close to neutral, it's going to carry a positive or a negative charge?
Positive.
Yes. All right. So lysine. What about arginine? Is it basic or acidic amino acid?
Basic?
Yes. So you have lysine and arginine are basic amino acids. You don't have histidine. If you have histidine, it's also basic amino acid. So these would tend to be positively charged at around neutral pH. So whereas aspartic acid and glutamic acid, from their name, they're acidic. So then at neutral pH, they would tend to carry a negative charge, right? What about glycine? What charge does glycine carry around neutral pH?
No net charge?
No charge, potentially, theoretically, right? So for simplicity matter here is just, I'm illustrating that the composition impact in a charge. So if I'm just looking at in a simple manner, I'd say, okay, I have 9 positive charges and I have 13 negative charges and 11 no charge, basically. So 9 will cancel 9, 9 positive will cancel 9 negative, then I'll end up with a -4 net charge. But this is a very simple peptide. It's not like that. You have so many, your proteins have a lot more amino acids, obviously, and then the environment, and it gets really complicated. We don't usually calculate it that way. We have systems that measure net charges for us. Okay.
Pam, I had a question.
Yeah.
When we approach this question that we solved, we should know the isoelectric point and then compare whether pH 7 is above or below its isoelectric point.
No, no. I don't expect you to know the isoelectric point of every amino acid. If I wanted to be, if I want you to answer based on this information, I'll give it to you. Here it's just take it for simplicity manner. I'm just illustrating the amino acid composition impact, not charge. And if we just assume that around neutral pH, your lysine and arginine will be positively charged because they're basic, and your aspartic and glutamic will be negatively charged because they're acidic. And then how do you calculate net charge? So it's basically just to illustrate that the amino acid composition impact your net charge.
I don't want to complicate things for myself, but I just want to clarify this again. If the isoelectric point of, say, lysine was 9 and a pH 7 is lower than 9, so then lysine should be negatively charged because...
No, positively charged.
Positively. Okay.
So under the isoelectric point, the proteins are mostly positively charged. Over the isoelectric point, they are negatively charged. Think about it, that what does it mean over the pH of the isoelectric point? That means you have more alkalinity. So then you lose protons. Protons carry positive charge. So when you lose protons, then you end up having negatively charged. In an environment that's acidic, you have a lot of protons in the acidic environment, then the protein starts picking up the protons, then it becomes positively charged.
Got it. Thank you.
Does it make sense? Okay.
Yeah.
All right. Good. So I'm just going to give you a 5 minutes break. We'll come back close to 3:50, maybe 7 minutes break then, and we'll start protein analysis. Okay, let's regroup. All right, great. So I'll share my screen again and move on to a new topic. So protein analysis. This is an important topic because it sets the stage for when we compare protein structure to function. It also sets the stage to understand production of protein ingredients. So it's really important that we cover this topic now. It has a lot of elements in it, and it is divided in to three categories: quantification, isolation, which isolation can be done for different reasons, and we'll talk about that, and then characterization. And characterization can be understanding the structure, which we'll talk a lot about here, but also you have characterization of functionality, where we talk about it when I introduce functionality. We will, for every functionality, I'll explain it, and then I would provide a methods of analysis. But for this particular section, we will focus on quantification, purification, and isolation for different purposes, and then characterization of structure. Okay, so again, think about this protein course as a puzzle. So we learn pieces and then we put them all together at the end and understand why do we need to know these different pieces. Okay. All right. So let's see. Some of you knows this. Some of you know this. So sorry. Some of you do not know this. So it's okay. We'll cover this together. But what do you know about these chromatographic techniques if they can be used to separate and isolate proteins in solution? Okay. So I see chat. Let's see. Okay.
All about.
Yes, we all agree. That's great. So I hope all of you have some background in chromatography or remember or have taken it somewhere, because I will brush over it today, assuming that you're familiar with it, given that you have a food science background, most of you, and those of you that don't already are taking food science classes. So chromatography has crossed your path before. So I will be mentioning it. I'll tell you how it's used, but I won't go into it in depth as I would go in food analysis class. I've spent 5 lectures on chromatography. We don't have that luxury here. So I really rely on you reading up and refreshing your memory. When I talk about chromatography, you can go back and read about it. I also put resources on Canvas for you to kind of refresh your memories. Okay. The following analytical techniques can be used for structural elucidation of proteins. Elucidation meaning just identifying the structure, determining components of the structure of the proteins. Any idea? Any brave soul is going to tell me something. Okay, I see. Who's the brave soul?
Is it A and B? So D.
Is it none of the above?
It is actually just one of them. All right, so N-terminal sequencing, I didn't expect you to know this. We're gonna talk. That's why we're teaching this, right? So N-terminal sequencing is usually used to determine, it's a procedure that is used to determine the sequence of amino acids in a protein. And I do have, by the way, documents on Canvas that talk about different unique methods. So you're going to find something on terminal sequencing, a summary about it, about MALDI, for example, or about FTIR. So there are documents that kind of summarize these procedures that we're going to talk about. It gives you a little bit of a background, more than I would cover here. Okay, so N-terminal sequencing, it shows us one component of the structure, which is amino acid composition. Amino acid, yes, composition, but the sequence, what comes in a sequence of protein. ELISA, which is immunosorbent enzyme-linked immunosorbent assay. Gosh, I kind of blanked out. So the ELISAs usually are based on detecting antibodies, linking antibodies with antigens, so specific antigens and specific antibodies. It's good for identification of antigens, sometimes semi-quantification or quantification if we have standards. So it is immunoassay. It's not necessarily telling us structure. It's telling us interaction of a protein with an antibody. Is it interacting? Is it not interacting? Is it immunoreactive? Is it not immunoreactive? So it's not really structure information. Western blot is also an immunoassay. So in Western blot is basically you run your proteins on a gel, and then you transfer them onto a membrane, and also you apply antibodies, and if you have a protein of interest that you're looking for will interact with the antibody, then you add the substrate to the enzyme that is linked to the antibody, and then you get a color, and then you would see if there is immunoreactivity or not. So again, it's an immunoassay. So it's not necessarily a structure elucidation, it's identification, determining the immunoreactivity, determining presence of certain type of protein. So that's what it is, okay? So we're gonna talk about differentiate these different protein analyses and what the purpose of these are. So when we hear the word protein analysis, it means so many different things. So it doesn't necessarily mean quantification. Quantification is just part of it, right? So you have quantification of the protein. We analyze proteins, we separate them. It's part of the analysis. So we isolate the protein, we separate it into a purified fraction. We characterize it and we identify it. So we can characterize it by looking at structural characteristics, or we can do identification using immunoassays, for example, or we can do liquid chromatography coupled with tandem mass spectrometry. So we hydrolyze the protein into peptides, we look at the library and determine what that protein is. If we don't know what it is, we can compare it to library sources, identify variants, identify different subunits. Ingredient preparation. So we do separation and purification. We can do it for analytical reasons, and we can do it for large-scale production of protein ingredients. Basic research. So we purify and separate the protein, and we look at its structure. We look at impact of processing on the structure. We look at impact of different purifications on the structure and functionality. So basic research, that research that will lead potentially to identifying industry feasible protocols to produce ingredients, identifying applications for specific ingredients, protein ingredients we're talking here. That's the industrial applications, basically. Okay, so there are different methods that we use and procedures, and we're going to talk as much as we can on different methods and procedures. But these methods and procedures, especially if there's going to be food application at the end, have to be legal, contribute to nutritional quality, and safe. And we think of economy, so cost effective, to be an industrial feasible application. And also when we isolate or when we process, when we test the proteins, we also look at how did everything impact the functional properties of the protein, because that's what leads to utilizing them in different applications. Okay, so this is an overview, right? I'm just going to go through this quickly because I'm assuming that all of you are familiar with protein quantification methods from previous classes. So we do have the common methods that we use for protein analysis is Kaldal, and the other one is DUMAS. Both are based on nitrogen determination. Infrared spectroscopy, IR. So we kind of determine protein content using mid-IR or near-IR after it's been calibrated for certain food products. And then we have research methods that we use quite a bit. BCA, bisynchronic acid assay, is based on the reducing power of the protein. So reducing the cupric ion to cuprous and looking at changes in absorption, and we run it alongside a standard. Buret and Lowry are different versions of methods based on reducing power of the protein. Bradford is an assay based on dye binding. Dye binds with the protein to give you a color. And then UV absorption is very simple. If you have a really pure protein and you just want to look at concentration and you know that you don't have any other interfering substances that may absorb at 280. So you can also measure the protein, but all of these research methods require running standard and having a standard curve, basically. So the next slide has a summary on these methods that kind of summarizes their principle, sensitivity, and so on. But before I go to that next slide, why do we care about protein content? Can you give me examples as why do we want to measure protein content?
For labeling purposes?
Yes, that is very true. One of the most common reasons proteins are present on the label, and we need to quantify whether it's a protein ingredient or whether it's a protein within a food product. Okay, and we usually use official methods of analysis, Kaldal and DUMAS. What else can you think of?
For running any sort of structure or function work, because a lot of it's based off of the weight of the protein that's in there, or the protein concentration, which isn't just the weight of what you're weighing out.
Absolutely. So when we're characterizing a protein ingredient, we really need to run our assay based on protein content. So the first thing we do is we determine how much protein we have so that we can set up our assays. Some assays are functional characterizations, some assays are structures, some assays are activity. So if it's an enzyme, for example, you want to know how much protein you have so you can determine the activity based on a gram of protein. Okay, great. What else can you think of?
Adulteration.
Say that again, Noah?
Adulteration.
Adulteration, yeah. If you want to make sure that you have protein, not melamine, so you want to use a method that quantitate the actual protein content. Yes, so let me show you. Oh, I'm clicking the other computer. All right, so nutritional labeling, where we use official methods of analysis, Kaldal and DUMAS are official methods of analysis. Quality control, when we're running a process, we want to determine the protein content alongside during the process to see that we're not losing protein anywhere. We are maintaining certain component, a certain fraction of proteins. So a quick quality control. Protein fractionation and purification. This is very important because especially when you're producing ingredients, you want to know what's your yield, how much of the protein you were able to extract. If you're starting with an X material, you want to get out as much protein as you can because protein is expensive and it's money. You don't want to leave anything behind. You will, because you don't have 100% rate of extraction, but it's very important to determine the yield and also to determine the purity of your final product. We talked about functional properties based on amount of protein and also biological activity. Okay.
Pam, I have a quick question.
Yes.
At 280, do other components of foods absorb, or is it just?
Yeah, so if you have polyphenols, for example, they might absorb at different UV. It might not be their optimal wavelength, but they might absorb at 280. So if you have a protein solution that you think might have other phenolic compounds that might absorb light, because the ones that absorb at 280 are the phenolic amino acids. So if you have any other phenolics in there that can absorb at UV, it might interfere and you might get overestimation. Does that answer your question? I don't see you, so I can't hear you either. Does that answer your question?
Hear me?
Now I can. But I didn't hear if that answers your question or not.
She says, I think she says you answered her question.
Okay, good. Because when I'm sharing my screen, I only see a couple of people and I don't see everybody else. Okay. All right, so this table here is kind of summary of the different methods, what you really need to know about these different methods. So, you know, Keldar, I don't expect you to remember the exact procedure, but you want to know it is based on measuring nitrogen. It's a chemical assay. It involves quite a bit of steps: digestion, neutralization, distillation, and titration. Okay, but I don't expect you to know the detail of the method. Nitrogen combustion, you use nitrogen analyzer based on combusting the sample and measuring the nitrogen content. And then from these methods, most common that we use in terms of research, absorption at 280, absorption at 214 is common for peptide bonds. So if we're running, let's say, on HPLC, a mixture of proteins and peptides. If you have peptides in a hydrolysate, they don't necessarily, may not contain tyrosine, tryptophan, and phenylalanine. A peptide might not contain that, but it will not absorb at 280, but will absorb at 214 or 220, in that range. 210 to 220 is, you know, wavelengths to look at peptides. Lowry and BCA, both are based on the reduction power. BCA is more common. We use it a lot in our lab. There are interferences. So for example, if you know that you have reducing sugars in your isolate, you will potentially, or any reducing agent other than the proteins, you would potentially overestimate. So, you know, these methods, that absorption at 280 or 214, Lowry and BCA and Bradford, are really meant for purified protein solutions that does not have interfering substances, because it's based on measuring absorption at the end of the day. The OPA here is listed. It is used for amino acid composition analysis. When you break down, when you want to determine amino acid composition, we usually hydrolyze the protein and run it on reverse phase HPLC, but Not all of them absorb light, so you react them with either an anhydride or OPA, which basically binds with the amine group and give you a fluorescent. ELISA can be used for detection and quantification if you have the right standard to produce a standard curve. Okay, so it's kind of like summarizing, be familiar with these methods, know where we use them, but I won't specifically ask you to describe a method. Okay. All right, so that's quantification, just getting total quantity. And then I want to spend some time on protein separation and purification because it is important in the protein world. Why is it important? What is protein separation utilized for? Why do we care about it?
Is it to separate protein and then study their interactions, like separate protein, isolate interactions with other compounds?
Okay, so basically isolate the protein and study them, determine their chemistry, their structure, their functionality. So studying the protein, identifying them, studying further, studying their interaction. If we want to purify an enzyme, for example, or if we want to produce a substrate for an enzyme. So we need to purify. Okay, that's good. What else?
Use the protein for products.
Yes, exactly. So produce protein ingredients. Okay, so production of protein ingredients, purify for further characterization, or for utilizing in assays, purifying enzymes, purifying enzyme inhibitors, purifying proteins that can be used as substrate in enzyme activity assays. So, yep. And another important one, and I'll give a lot of examples on it later, is determining the impact of processing. So when you process, whether it's, let's say, flour or any, you're milling a flour to extract protein, or you are heating the flour to inactivate, to pasteurize or inactivate pathogenic bacteria. So you need to treat it, or you are heating soy flour to inactivate trypsin inhibitor. So what happens to the protein that you potentially might want to use for something else? I'll give you an example about heating soy flour that is used for fermentation purposes and growth of yeast. And changes in the heating properties impacted the protein digestion by the yeast. So I'll give you that example. So impact of processing, whether it is processing to formulate ingredient or when we are formulating with the proteins, what happens to the protein structure, functionality, and nutritional quality. So in order to understand how we can separate proteins and isolate them, we really need to know what specifics of these proteins that will allow us to separate them from a mixture. So we know that proteins differ in nutritional quality, they differ in functionality, but they also, the most important characteristic that we will rely on for separating protein is the biochemical types of characteristics. So the biochemical types of characteristics, meaning so solubility under different conditions that will rely on charge and hydrophobicity, for example, of the protein. How stable to heat are they? Because we can separate protein based on denaturing them and precipitating them out of solution. How big are the proteins? What's the size? We can separate them based on their size. We can separate based on their configuration, their structure. And we can separate based on surface properties that allow them to adsorb to different surfaces and their affinity to different molecules. So these are all biochemical differences that is impacted by the composition of the protein, the sequence of the protein, amino acids within a protein, and the final molecular structure. So for example, to separate away from casein, there are two different types of proteins. They have different solubilities. And then they're impacted by pH and salt differently. And also they are impacted by centrifugation force differently. So we can separate casein from whey protein in different ways. This is a structure of, a simplified structure of a protein in soy. So the quaternary structure also determines how we can separate them and if we want to separate different subunits. So these circles are individual subunits. If I want to isolate a particular subunit, I want to understand what interactions is going on within this molecular structure that allows me to separate these black circles, particular subunits, from those blue circles so I can study these individually. So actually, this is a project that Holly will be working on for pea protein, trying to isolate particular subunits and looking at the impact of these specific subunits on the functionality. So we need to understand a lot about the biochemical properties so we can actually isolate these proteins. So special consideration when we're isolating proteins. So what else do I need to know and consider when I start with any material that I want to take out the protein? So I will potentially lean towards my students that already isolate proteins. What do we care about and think about before we start isolation? Can anybody think of something?
Competing molecules?
Competing molecules.
Like phenolics?
Okay, yes, we want to make sure that we, let's say, don't oxidize phenolics and cause flavors and browning while we're isolating the proteins. Okay, good, that's one thing.
I think we need to think whether we need to pre-treat the raw ingredients first, either by degumming or defatting.
Yes, so looking at the composition of our starting material. So is it high in fat? So I need to think about defatting. Okay, is it high in gums like camelina? For example, we need to remove the gums so that we are able to extract protein and filter, and these gums don't ruin our filters, for example. So yes, so the composition of the starting material. So let me reveal some of this. Okay, so the nature of the food matrix, it's very important. What do we have? Interfering substances, fat. We need to remove it before we isolate. We don't want to form emulsion during our wet processing to remove protein. So we need to remove the fat so that there's no interference. And I can extract as much protein as possible. So how much purity am I looking for? Am I looking to produce a concentrate? Am I looking to produce an isolate? So a concentrate could be anything less than 80% protein, and an isolate above 80%. Sometimes to call it an isolate, it should have more than 90%. So different procedures are used to get different purities. Also, the process that I use, how will it impact the functionality of the protein, whether it is an enzyme, how is it going to impact biological activity, for example. So some proteins are not soluble at neutral pH. Some oilseed proteins require really high pH, and I'll show you examples later. To solubilize, great, I can solubilize them, but now high pH also denatures my protein and causes oxidation and causes polymerization. Then the protein might not be very functional at the end of the day. So I need to look at other ways to isolate that protein. Cost is very important for industry. What kind of procedure can be scaled up so that it does not involve a lot of waste, a lot of use of energy, a lot use of water, a lot of use of chemicals, So I need to find the most appropriate method of isolation that would be cost effective and scalable. How do I monitor effectiveness of my isolation? What do you guys do, those of you that isolate proteins? What parameters you look for?
Ash moisture, we measure approximate analysis to measure the ash moisture and protein.
So protein purity. So one way to measure your effectiveness of your isolation and to determine the purity. Ash is very important because you want to make sure that you don't have really high salt, because salt impact the functionality of the protein and the final application. Moisture-wise, it's important. You want to make sure when you produce a protein ingredient, it is dry with 10 to 13% moisture or lower, because for storage, you don't want microbial growth. Okay, good. What else is important? When you isolate your protein, what do you measure as well?
Your yield.
Yield, yes. Yield is important because it's related to cost. Am I able to get all the protein out? So total quantity, that means purity. Yield, what happened to the protein? Again, is it still structurally viable and functional? So these are important parameters. I didn't have salt in there, but it is important. I didn't have moisture, but it is important too. Okay, so here's another table that summarizes the process of purification. So what do we start with? First of all, we start with extraction. So we need to remove the protein from a matrix. So we extract it. We can solubilize it, for example, is one thing, to separate it from others by specific conditions that would solubilize the protein. And you can, if it is a starchy material, precipitate the starch. If it's a fibrous material, precipitate the fiber and solubilize the protein. So it is basically having different phases and different solvents that will help you get the protein into solution. But then in that solution, you might have other components. You might have sugars, you might have soluble fiber. So you want to purify it further. So you take the characteristics of the protein and say, okay, I'm going to change the environment so that I can precipitate the protein and separate it from solubles such as sugars and colors and soluble fiber. So you can purify it even more. Instead of precipitation, you can say, okay, I'm going to separate it from small molecular weights, sugars and salts and colors by membrane filtration. So we'll talk about that. You can do membrane filtration, applying pressure so that you can concentrate and remove other solubles that you don't want in your final product. You can also separate by ultracentrifugation. For example, caseins are precipitated from whey by ultracentrifugation. Or you can separate proteins using chromatography, and it is really widely used in industry. It is used actually, a very common example is production of whey protein isolate. So Davisco, which is now Agripur, they are unique in the way they produce their isolate, not by membrane, but by ion exchange chromatography. They isolated and separated from lactose and salts and concentrate the proteins in whey that way using chromatography. Other types of chromatography are also used: size exclusion, reverse phase, hydrophobic interaction, but some of these are used for research purposes more than industrial purposes.
I have a question. So is the whey protein isolate made from ion exchange different from a membrane process one?
Of course, they differ in composition. In the membrane, you have higher calcium content, and you have lower beta-lactoglobulin to alpha-lactalbumin ratio, and you have different degree of denaturation. So you do have different characteristics of the isolate in terms of protein composition, salt composition, and lactose composition. You have higher lactose content in a membrane filtered WPI versus an ion exchange WPI.
Okay. And do they got to specify if it's ion exchange made or if it's done through membrane processing?
Yes, so you would know, well, I don't know if they say on their label of the ingredients, but we know that Agripur is ion exchange product and Galambia is membrane product. I mean, I know, I don't know if they probably put it in their specification sheet or something.
Okay.
Yeah.
Thank you.
Good. So this is kind of a summary table. Again, good to study and to have a general idea. But I'll walk you through the protocols of purification, the main ones that we either industry utilized or research utilized. Okay, so nowadays there's a common process for pulses. So there are a lot of pulse ingredients out there now that are gaining traction and attention in the industry. Pea protein and chickpea and lentils and beans. The main processing for purification of these proteins is dry milling process. So with the dry milling process, it is basically air classification. So they spin the powder, the flour. So let's say in pea, you get the split PUD hull, you split the pea, and you mill it into flour, and then you get that flour and you put it into an air classifier. You separate it based on density. And what you get two fractions out of it. You get a fraction that is high in protein, and it has some starch and fiber, but it is about 55 to 60% protein. And then you get another fraction that is rich in starch and fiber and has less protein in it. So you have two fractions. The fraction that is rich in protein is called protein concentrate and is utilized in different applications. But it cannot purify, this process cannot purify the protein further into an isolate. You cannot get a 90% protein purity from dry milling. So if you want to get a really high purity protein, we need to go ahead with something we call wet milling, where we try to solubilize the protein and isolate it in a way to get above 80% purity. The most common one in industry is pH alkaline extraction. This is the most common one that is used for plant proteins, basically. So it is used to first solubilize the protein. If you look here, this is soy solubility curve. You see at alkaline pH above 7 towards the 8, you have really high solubility. So if you solubilize at pH 8 or 9, you get most of the protein out of the soy flour that you defat first, for example. I'll show you a diagram in a minute. But then you have soluble fibers and you have sugars, oligosaccharides, colors. You don't want those in your isolate. So you have your soluble proteins and other solubles, so you tend to, they precipitate at the isoelectric point of the protein. They change the pH of the solution to where the protein precipitate and other solubles remain soluble. And then you get the precipitate out and neutralize, pasteurize, dry. So you get an isolate that way. Another wet milling is salt solubilization. So with salt solubilization, you have, okay, you have your flour of whatever matrix it is, devoid of fat. First of all, you have to make sure you don't have high percentage of fat. Then you put that sample in a low concentration of salt. Then you salt in. So in this case, you have some salt interacting with the protein and increasing the charge load, meaning increasing protein-water interaction. So you solubilize. And then along with the protein, other solubles. So the next step is you want to remove that protein from the other solubles. So there is the salting out. So I'll talk about salting out in more detail, but you increase the concentration of a specific salt, usually ammonium sulfate. It kind of shields the charges on the protein, prevent the protein from interacting with water, and then it aggregates and precipitates. But this process is not used a lot, and it's not used in industry, period, because of the use of high salt. And then you need to wash that salt out. So you're going to use a lot of water, and then you will generate a lot of waste. So instead, what's been investigated is instead of removing the precipitating the protein to remove it from solubles, small sugars and salts, what you do, you can do membrane filtration. So after salting in, you kind of concentrate your protein by removing the small molecular weight components using membranes with particular pore sizes. So this is kind of an overview of plant-based protein isolation. I'll give you an example of whey later on, whey protein. So if we want to separate based on differential solubility, here's I'm going to go a little bit into detail about salting out and how that happens. So mostly ammonium sulfate, and ammonium sulfate is a salt that interacts strongly with the surface of the protein and shields its charges. So instead of the protein interacting with water, it then has attractive forces between the molecules of the protein because they're no longer interacting with water, and the charge is shielded. You don't have repulsion, then the protein aggregates and precipitates. So here's a question for you. And again, think in very simple terms. I'll show you a complicated example later on. But here, if I have 5 proteins and they differ in their net charge at a particular pH, where E has the highest net charge and A has the lowest net charge. And if I want to separate C from this mixture, what do you think would be my approach? What do I do? Think a little bit about that, but simplify it in your head. Think about salting in, salting out. What would you do? I'm not looking at a very detailed approach, a general high-level approach.
Something like a 2-step separation where we would first get rid of A and B by using a certain amount of salt concentration. And then you add salt in a way that, and then you have a mixture of C, D, and E basically. And now in that mixture, you add high concentration. So C, or just try to vary in a way that C remains in the solution and D and E are taken out.
You're very close, and very, very close. You just messed it up a little bit at the end. So it is a 2-step process. We really need to know the characteristics of A, B, and C, and D anyway, but in simple terms, I start by a low concentration of salt that precipitates A and B, because A and B are less charged. So they will require less concentration of salt compared to C, D, and E to precipitate. Then I get the precipitate, get rid of it, get the supernatant. Now here, Radhika, where you would add a salt a little higher than you used for A and B, that would only precipitate C and keep D and E in solution, then you would retain the precipitate in this case.
Got it. I got confused between the precipitate and the retentate.
Yes, but you were very close. Good, it's good thinking. So but in order for us to really know what concentration to use and what salt to use, it requires a lot of experimentation and a lot of knowledge about our proteins, their characteristics, their charge, how they're impacted by pH, by different types of salt. So a lot of research, and luckily for us, it has been done for a lot of proteins. So there are established methods that for many different types of proteins that help us isolate. But there's always room where, oh, it's a new type of protein. We just have to study it and experiment a little to isolate it. So separation of solubility also by precipitation with using the isoelectric point, which I talked about earlier. During wet milling, you solubilize and then you use the isoelectric point of the protein. So basically the protein loses its charge. It no longer interacts with water. It's attracted with each other because you no longer have a net charge. You have hydrophobic interaction. You precipitate the protein at the isoelectric point. So that is another way of precipitating the protein. You can precipitate the proteins with solvent. So a solvent is an organic solvent, so hydrophobic residues like organic solvent. So when you add, let's say here, acetone, for example, it is going to interact on the surface of the protein, and now the residues in the inside of the protein, they're hydrophobic. So they're now, oh, there's acetone. I can just open up a little bit and interact with the acetone instead of interacting with the water anymore. So then the proteins now interact with each other via hydrophobic interactions because the hydrophobic groups are exposed now. So you can precipitate the protein that way. Another way of precipitating the protein is denaturing it, which is the least favorite way if you want to produce a functional protein. But it is a way. You denature the protein. For example, you can separate whey protein from casein by heating up. The whey protein will precipitate because it denatures at higher temperature than casein. Just because the question here, why? Because it has a globular structure. Every ordered structure is impacted by heat more than a disordered structure. So when you have an ordered structure, it is impacted by heat more than a disordered structure. So casein denatures at really high temperature. It has a very disordered structure. Whereas beta lactoglobular in this case is very ordered, so heat will denature it more readily. So denaturation exposes hydrophobic groups, then hydrophobic groups will attract each other, and when the proteins are close to each other, if there are disulfides or if there are sulfhydryl groups, SH that is, then you have them in close proximity. If you have the right pH and heat, you can form disulfide linkages and even cause more precipitation and denaturation that is permanent. That's why this is not a favorite approach to isolate a protein to remain functional. Okay, here's the example that I told you. I'll show you. So this is for isolation of protein from soy. And we start with, so the soy usually, I'll show you the diagram of the entire thing when we talk about soy in particular, but the soy is dehulled, the soybean, then it's cracked and flaked and pressed so that you remove the oil and then you defat it further by hexane and you end up with defatted soy flakes. So then you have two routes, either produce a concentrate or produce an isolate. To produce a concentrate, you can add 80% or 60 to 80% of ethanol, alcohol. So you solubilize sugars and oligosaccharides and other solubles, and the protein and fiber, you don't have much starch in soy, protein and fiber will precipitate. And then you take that, you pasteurize, dry, make sure it's neutral, resuspend, and you get soy protein concentrate. You can get the concentrate if you take the flakes and also precipitate at acidic pH directly. So take the flakes, put it in water, and then acidify. Then again, the protein and fibers will just precipitate, and you can get rid of the solubles as well. So you end up with this concentrate, and it's often used to produce texturized vegetable protein. The other route where you actually produce an isolate is you take the flakes, obviously they have to be melted into flour to enhance surface area for extraction. You use alkaline pH 7.5 or higher, and you solubilize the protein and other soluble fibers, soluble sugars, oligosaccharides, and precipitate the insoluble fiber. Then you take the supernatant and now you acidify to the isoelectric point. And from there, you separate it from oligosaccharides, other sugars, the colorants, and you get a precipitate that is mostly protein. It has some salt in it associated with it. So you take that precipitate, separate it from the solubles, but then you resuspend it and neutralize. You never want to dry your protein at the isoelectric point. You cause permanent denaturation. So you redisperse, neutralize, and pasteurize, homogenize, and spray dry. And you get your isolate, which is over 90%. You can get more purity if you do ultrafiltration at the end. Ultrafiltration, diafiltration, you can concentrate, remove salts, and you get even more pure isolate at the end. So that's kind of a way to produce concentrate and isolates from soy. Separation now by adsorption. And this is where I gave you the example of the whey protein by agropore. So they use actually anion exchange and chromatography. And in anion exchange chromatography, again, refresher to remind you, we're exchanging anions. So your proteins are negatively charged and your column has media that is positively charged. Then the anions are separated based on how negatively charged they are. So we can do that in the lab for research purposes using analytical columns if we want to isolate the proteins to look at any characteristics and differences among the proteins, or we can use them in a semi-preparative way, or it can be industrial, like large-scale columns that can use that are used to isolate the protein. And usually the media that is used to isolate, that you fill the column basically, is polysaccharide base because it's proteins. It can be different bases if it's smaller molecules, but for proteins specifically, it is polysaccharide base because you need larger pore size for the proteins to move along the column. And the hydroxyl groups are derivatized. That means they can have a functional group that is either negatively charged, weak acid, or positively charged, weak base. So cation exchange will have a negative charge in the column. So cations, that means positively charged molecules or proteins, will be separated. Whereas when we're doing anion exchange, you would have a base that is positively charged and your proteins that are negatively charged will be separated. Again, this is a review for you. I'm not gonna, again, chromatography can take 5 lectures to describe, but this is a refresher and we have reading material to help you remember chromatography. Hydrophobic interaction chromatography is very common in the protein world, and we use it for different purposes. But let's say if I have a mixture of carbohydrate and protein, it's It's one of the easiest ways to just get the protein separated from a carbohydrate. And we apply that in the lab. And here's how it works. So your media or your stationary phase in the column has hydrophobic groups. So the proteins interact with them via mild hydrophobic interactions. To enhance interactions with the protein, we usually put the protein in salt solution. So it kind of masks the charge, and then the protein can interact via hydrophobic interaction. Then if we have carbohydrate, they're not going to interact via hydrophobic interaction. So they get washed. When we have salt solution running, the carbohydrates will get washed and the proteins will retain on the column. Then I sift to water, theoretically, then the protein will lose the salts on it, and then it will get back its charges. Then it's no longer interacting with the column and it comes out. So then you have your protein in the water phase. You can separate different proteins in a mixture by building a gradient with different salt solutions. Then the most hydrophobic will remain the longest on the column. The less hydrophobic will go faster, and the least hydrophobic will go first. Then you can separate based on the hydrophobicity of the protein. But if you're just, for example, trying to get the protein separate from carbohydrate, then you don't really worry about gradient elution. So just a few minutes here, like I'll finish at 4:50. I'll just continue with this a little bit longer, 4 minutes. So affinity chromatography is also used, and it's used a lot when we want to isolate antibodies to develop ELISA assays, for example. So it is used, affinity chromatography is used. So the protein here could be the antibody, and then the specific antigens could be the functional groups in your stationary phase or your media that you're using. Other way around, you can specifically isolate an antigen, a specific protein of interest, if you have a specific antibody linked in your media. So it's very commonly used for antibody-antigen purification and also for enzyme purification. So if you have an enzyme that you want to purify for a specific assay, so you can put enzyme inhibitors as the functional groups on your stationary phase or media, and then your enzyme will interact with them and get separated from the rest of the proteins and components that way. Glycoproteins are often separated if you have lectins as functional groups. Now, once you have that protein of interest very specifically bound to your media, you want to get it out of the column after everything else leaves the column. What you do, you change the environment, change the pH, the ionic strength. So you kind of weaken the interactions, and then that particular protein can elute. Or sometimes you add the specific functional group that is on the column, you can add it to the mobile phase and also cause that particular protein to interact with your solvent or mobile phase moving down the column. Okay, so separation by size. I guess, I don't want to start this now because we have a bunch of separation by size. I'm going to stop here and pick up next time. So far we've finished separation based on solubility and binding to different surfaces, adsorption and solubility. Now we're going to talk about size, but not now, next Wednesday. Okay. I will stop here and have a great evening, everybody. And I will see you next Wednesday. Come with questions. And nobody interrupted me for any questions, maybe a little bit. But next time, come with questions. I'm happy to, have a discussion. All right. See you later. Bye-bye.